Hello biotechnicans, welcome to today's video where we will be discussing about a very important topic from CSIR NET Life Science syllabus, that is comparing oxidative phosphorylation and photophosphorylation which take place in plants. We will be looking at the differences, similarities, important components, characteristics and significance of both the processes. Let's begin. I want to remind you that there are other entrance exams also that include this topic in their syllabus. So in any case, any of these aspirants are watching this video, do watch it to the end. It is going to be very helpful for you. That is CSIR NET Life Sciences, GATE, BT, Excel, ICMR, TIFR, ICAR and DBT as well. Now let's first understand the very basic steps for oxidative phosphorylation process. We will be trying to look at it in very simple steps. We know that when we consume some food like any macromolecules in the form of carbohydrates, fats or proteins, they will be broken down and they will be oxidized to give rise to reduced equivalents. Now what do we call those reduced equivalents? They are NADH and FADH2. After getting the electrons, they will become reduced. So what you are seeing here, NADH and FADH2, these are the reduced forms since they have the electrons in them. These electrons will now be delivered to the electron transport chain, where it is consisting of a series of complexes present in the mitochondrial membrane, which will be taking the electrons from the NADH and FADH2. Okay, so this is the first step here in oxidative phosphorylation. Electrons are transferred to electron transport chain, since the NADH and FADH2 will already be giving the electron, that is why NAD plus and FAD will be regenerated so that they can again enter another round of oxidation steps. I hope everyone is aware of the glycolysis Krebs cycle. This is how the macromolecules will be oxidized to generate electrons, which are then carried by this NADH and FADH2. Now what about these electrons? What will happen to these electrons? As I said, the electron transport chain, it is consisting of a series of complexes where one carrier to another carrier, the electrons will be transferred. Okay. Now very important point over here is that the flow of the electron through the electron transport chain is downhill. What do you mean by downhill? Means the electrons flow from a region of higher concentration to a lower concentration. That's what is generally downhill. But in this case, it means that they will flow from a lower redox potential to a higher redox potential complex. What is redox potential? Redox potential is the capacity to get reduced. That means the capacity to accept electron. So when we start with the initial electron carriers in the electron transport chain, they have less capacity to accept electron. But as we go down the electron transport chain, as we go further towards the end, the carriers which are present towards the end, they will have more capacity to accept electrons, which means they will have higher redox potential. Okay, so that's why this particular process of electron transport is favorable or it is spontaneous. It happens on its own just because it is becoming more and more easy to give away the electrons, so it doesn't consume any additional energy. So this process is what is spontaneous. Whenever any process is giving out energy, that energy will be used to drive some other unfavorable process. So this another unfavorable process is H plus pumping. Whenever the electrons are passed on from carrier to carrier in the inner mitochondrial membrane, some protons which are present in the matrix side of the mitochondria, they will be transported across the membrane. Now the inner mitochondrial membrane of mitochondria is impermeable to these H plus ions. Hence H plus ions cannot just directly cross the membrane. They have to either get pumped through the complexes, and if they have to come the other way, they will do so through some proton channel which is present in the ATP synthase enzyme complex. That's why the hydrogen ions pumping will take place through the electron transport chain complexes, and this will happen with the energy which was released from the downhill electron flow. Because when you have the H plus coming to the other side of the membrane, then there will be an abundance of H plus ion which is of higher concentration. Now, if you try to pump something from a low concentration area to a high concentration area, that will obviously need energy. So, from where it will get the energy? From the energy which is released by transferring the electrons from in the downhill direction. Okay. Now, this particular H plus is very important in building up of a gradient. But before that, let me tell you that what happens to the electrons. Because it is passed on from carrier to carrier is fine, but ultimately it will be taken by something, right? So, there should be some terminal electron acceptor. In case of oxidative phosphorylation, the electron acceptor is oxygen. But we know that oxygen is present in the form of molecular oxygen is O2. That's why this oxygen needs to split up. Okay, so basically we will need half oxygen. So that's what it means. And this will combine with two H plus ions, and the electrons which we are getting from the end of the electron transport chain, those two electrons, they are given to oxygen. But oxygen is split up and it will lead to the formation of water. Since in water we have just one oxygen, that's why this splitting up takes place. So from one molecular oxygen you can actually form two water molecules. That's why it is written like that, splitting of oxygen to form the water, right? Now this H plus ions, they are present they are a different one which are in the matrix to form water. But right now the next step which I am talking about is the ATP synthesis, where I am talking about the H plus gradient which was created in the intermembrane space. As I said, the H plus ions are transported from the matrix side towards the intermembrane space, which is the space present between inner mitochondrial membrane and outer mitochondrial membrane. When H plus ions are transported into the intermembrane space, their concentration increases here, which leads to the formation of a positive proton gradient. And this proton gradient tries to balance itself by coming back to the matrix. As I said, it cannot do so by its own. It has to come through a F0, F1 particle which is present in the inner mitochondrial membrane. It is the ATP synthase. So from here you have a proton channel through which the H plus ions will come, and as it will do that, there will be a rotation in this particle which helps in the formation of ATP. So that is the last step, which is the ATP synthesis. H plus ions will pass through ATP synthase into the matrix. Let me show you the representation, diagrammatic representation. Now this is what is the inner mitochondrial membrane. Okay, we know that inner mitochondrial membrane, mitochondria, it is a double membrane structure, and the inner mitochondrial membrane is thrown into folds which are called as cristae. The reason is because they want to increase the surface area so that they can accommodate more and more of this electron chain complexes. That's why the mitochondria is called as the powerhouse of the cell, as it has many such electron chain complexes embedded in it. Okay. Outside the outer mitochondrial membrane will be inner mitochondrial membrane will be outer mitochondrial membrane, which is freely permeable to all the molecules, but inner mitochondrial membrane is impermeable to most small molecules. Okay. The space between outer mitochondrial membrane and inner mitochondrial membrane is the intermembrane space. So this is where the proton gradient is built up. Now what did I say? This process starts with NADH and FADH2. So that is a separate series in which FADH2, it will be given to another type of a complex which is not mentioned over here, just to keep simplified. So that is what is called as a complex 2. It is called as succinate dehydrogenase complex. We know one of the enzymes in TCA cycle is succinate dehydrogenase, which will be giving the electrons to FAD, because FAD is a prosthetic group of this particular enzyme complex. Okay, now when NADH and FADH2 gives the electrons to electron transport chain, they are transported to carrier. You can refer my another video which is on this respiration and photosynthesis, which I have discussed in another video, if you want to know the electron transport chain in detail. But here our focus is to compare these both processes. So when we have the NADH giving the electrons to carrier, as you can see, ultimately it will be giving to oxygen. As I said, half oxygen and 2H plus. This is when the protons are transported to the other side of the membrane. Okay, this leads to the formation of a positive proton gradient, which is electrochemical potential. Electro because this is regarding the charges, and chemical because it is regarding H plus ions. We know that the H plus ion it will be impacting the acidity pH of the environment. That is why it is called as electrochemical potential. Okay, now this side, that is intermembrane side of the inner mitochondria, basically the inner intermembrane space, it will be having excess of positive charge, hence it is referred to as the P side. Okay, whereas this matrix side, since it is now devoid of electrons, like lesser H plus ions are there, that is why this side is called as the N side. You should be very much familiar which is the P side, which is the N side of the mitochondria. Now that this H plus ions are built up, they want to come through the ATP synthase. As I said, ATP synthase is just like a rotor. Whenever the protons will pass through it, this rotates, and the part embedded in the membrane is the F0 particle, and the part which is facing towards the matrix, it is F1 particle. From here only ATP will be synthesized. Whenever the protons will pass through it, this will occur rotation. F1 is the component where ADP and PI will be attached, and with the rotation of this F1 particle, there will be a conformational change, thus leading to the formation of ATP. Okay. Now let's move on to the photophosphorylation steps. Photophosphorylation, it happens in the green plants, which is photosynthetic cells. Now here we use light energy. Light energy, it is used to break down water, which is called as the photolysis, to produce hydrogen ions, electrons and oxygen. As we saw in oxidative phosphorylation, that water was produced at the end by using electrons and oxygen. Here is a contrasting step, that in the start itself of photophosphorylation, the water is broken down to release the electron, and in fact this is the source of electrons. Well, while in oxidative phosphorylation, the source of electrons was from the oxidation of the food that we are consuming, or the plant which is consuming, and here the source of electron is water. Then a similar process with oxidative phosphorylation is that the electrons which are released from the photolysis of water, they will be traveling through the electron transport chain, just that the complexes, there are no complexes here, there are carriers. The carriers here will be different from that of the oxidative phosphorylation. So they are also present within the membrane. However, the membrane is not of mitochondria. Now we are talking of chloroplast, because this is the site where this particular process occurs. Okay. Now while the electrons are traveling, the same concept is there. A proton gradient will form, which will lead to the formation of ATP synthesis, a proton gradient. Same way the protons will be passed to the other side of the membrane. I'll show you which side. And again they have to come back through the chloroplastic F0 F1 particle, which is called as CFO F1 particle, CF1 particle, and ATP will be generated. And ultimately this is something which we don't see in oxidative phosphorylation, that the electrons will ultimately be given to reduced NADP. I mean, they will be given to NADP. Okay. So after getting the electrons and H plus ions, it will become NADPH. So what we saw in oxidative phosphorylation was that NADH was the source of electrons, and it was oxidized when it was starting the process, right? Oxidative phosphorylation, after giving the electrons, those were getting oxidized. But here The NADP is the final electron acceptor. After taking the electrons, it becomes reduced, and this will be utilized further in the dark reactions of photosynthesis in order to produce glucose molecule. Again, you can refer to my other video if you want to know details of this photosynthetic processes. Let me show you the photophosphorylation diagrammatic representation. As I said, this happens in the chlorophyll. Now, chlorophyll, you would be knowing, inside the stroma, it is a double membrane structure. So, inside the stroma, which is the lumen of the chlorophyll, we have some stacked molecules. So, this not molecule, fluid filled, some sacs, cavity filled sacs, which are called as these are called as thylakoids, isn't it? So, one of them is thylakoid, and the stacks, they are called as grana, right? Grana or granum. Now, the photophosphorylation happens in these thylakoids only. Now, what you are seeing over here, it is a thylakoid membrane, right? Now, thylakoid membrane, inside the thylakoid membrane, so outside is the stroma, and inside the thylakoid membrane, it is the lumen. So, here the electron transport chain carrier, so it's not called electron transport chain carrier, here we call it as photosystems, okay, photosystem 1 and 2, they are located in this thylakoid membrane. So, what you are seeing over here is the electron carrying system, electron transporting system, and here itself the F0 F1 particle is present, but the F0 F1 particle faces the stroma. So, ATP, it is released into the stroma. What we saw in oxidative phosphorylation, the ATP was facing, the F1 particle was facing the matrix side. So, ATP was released into the matrix of the mitochondria, from which it would have been, it has to be transported to the cytosol through some specific transporters. Okay, but here it is released in the chloroplast, in the stroma of the chloroplast. Now, what are again the molecule, what are the carriers that we have? I am not going into detail. We have this photosystem 2 and photosystem 1. From here, the carrier to carrier, it is given, ultimately the electrons are given to NADP+. Okay, so NADP+, after taking electrons, it will form NADPH. In this process, the electrons are donated by water. As we saw in the previous one, the electron source was NADH. Here the electron source is water. It will be breaking into oxygen and H+ ions, and the electrons will be given out. This is what is called as photolysis of water, because the energy comes from the photons which are absorbed from the sunlight, right? Now, where are the H+ ions sent across? So, H+ ions will be transported from the stroma into the lumen of thylakoid. So, this is where we have the gradient. Okay, so what will, where in this case, where will be the positive side? It is the lumen. Okay, so remember, in case of photophosphorylation, P side is the lumen and N side is the stroma. These all details are important when you are comparing these logics. Now, while we are talking about types of phosphorylation, let's also see what are the different types of photophosphorylation that we have. In a rough, quick overview, the photophosphorylation of ADP to ATP can be cyclic or non-cyclic depending on the pattern of electron flow in photosystem 1 or photosystem 2 or both. Now, what do you mean they are called as cyclic? Now, cyclic means only one photosystem is involved for the passing of the electrons, and in non-cyclic photophosphorylation, both photosystem 1 and photosystem 2 are involved. First, let me show you about the non-cyclic. So, from this is where we know about the photolysis of water, where electrons are released. These electrons are then sent over to the first reaction center. Okay, so here light is absorbed by the photosystems. In case of non-cyclic, both photosystems are involved and light is absorbed by both of these photosystems. Okay, it involves PS1 and PS2 both. That is the difference between cyclic and non-cyclic. So, I'll compare between these two as well. Now, light is absorbed by both the photosystem and it leads to photoactivation. What is photoactivation? Photoactivation, actually the light is passed through the primary pigment. There are some accessory pigments also surrounding it, to the primary pigment of each photosystem. Photoactivation happens, which means that electrons in the primary pigment molecule of each photosystem, they are excited to a higher energy level. So while they are excited to higher energy level, from here these will be transferred from these carriers. Kindly note that in CSIR the sequence of these carriers sometimes comes in a part C question. You have to arrange the order properly, what carrier to what carrier this will be transported. Okay. Now the excited electrons from PS2, they will be passed on to PS1. And while they are doing that, some protons will be transported to the other side of the membrane, which I said, and this will lead to the formation of ATP. However, that is not being shown in non-cyclic photophosphorylation. The focus over here is on the flow of electrons. Now, because of this H plus gradient is created, as I said, now the excited electrons from PS1, now that the electrons have come to PS1, even this is undergone photoactivation. It will go to an excited state. Then there is another set of carriers. You can see that the set of carriers here is different from here. So again the electrons will be given, and finally it will be given to NADPH, which is the end product of this non-cyclic photophosphorylation, which then enters the dark reaction. So in this non-cyclic photophosphorylation, NADH is also produced, and since this is also taking place from here, ATP is also produced. So both ATP and NADPH is produced over here. Okay. Now coming to the cyclic photophosphorylation, this is what is a cyclic. Yeah, you can see that photosystem 2 and photosystem 1 is written, but however this part doesn't take place. So cyclic photophosphorylation involves only PS1, not PS2. If you will see that from here, there is a coming back, okay, like there is a reverse flow of the electrons here, and that is in the form of a cycle. So this only keeps on cycling, right? Now what is happening over here? The light is absorbed by photosystem 1, which is in the thylakoid membrane, and passed to the photosystem 1 primary pigment. Then an electron in the primary pigment is excited to a higher energy level, and it is emitted from the chlorophyll molecule in a process which is known as photoactivation. This excited electron is captured by the electron acceptor, transported via a chain of electron carriers, and before being passed back to the chlorophyll molecule photosystem 1. That's why it is called as cyclic. As electrons pass through the electron transport chain, even they will provide energy to transport protons to the other side. So here also the H plus ions will be created. Okay, so electrons passing from P700 to ferredoxine, this is ferredoxine, do not continue to NADP plus but move back through the cytochrome. So as you can see from here, if it was non-cyclic, it would have gone towards the formation of NADPH, but instead from FD, ferredoxine, it comes back again to this part. So this is the part where ATP might be generated but not the NADPH. Okay, so here electrons are repeatedly cycled. They are not leading to the formation of NADPH, but they are cycled. Let's compare both of this, cyclic and non-cyclic. In case of non-cyclics, electrons do not come back to the same molecule, whereas in cyclic, electrons come back to the same molecule. In non-cyclic, first electron donor is water, because that is the source of electron, but in cyclic we are directly getting the electrons from PS1. PS2 gives it to PS1, so directly from PS1. Non-cyclic involves both PS1, PS2, cyclic involves only PS1. Last electron acceptor is NADP, but here there is no last electron acceptor since it goes in cycles. So last electron acceptor is again P700, which is PS1. The net products are ATP, NADPH and oxygen. But here in cyclic, as I said, NADPH will be not produced, and since we are not having the photolysis of water in cyclic, that is why oxygen is also not produced. This product will be only ATP, right? Now let me tell you that as I was saying, in CSIR exam, every little detail is important. If you are part of Biotechnica's classes, online classes, if you are attending them, then you will get to know how much depth and detail we go into. Okay, so current batches are running and even new batches are going to start up for CSIR NET life science exam preparation. In case you are a beginner and you are interested, then I will strongly recommend do attend these classes, because we teach every little concept from the very basic. Okay, now proton gradient in relation to ATP synthase orientation. Just now we saw in mitochondria, the P side was the intermembrane side where the H plus ion gradient was created, and the ATP was getting generated in the matrix, which was the N side. So whichever is the N side, there the ATP will be generated. Remember this. The statements in exam can be confusing in true or false, but you have to answer carefully. In case of chloroplast photophosphorylation, the thylakoid lumen is the P side where the proton gradient is getting generated and ATP is generated towards the stroma. So F1 particle is oriented towards the stroma. In bacteria also it happens in some of the bacteria. So the cytosol is the N side there, and the intermembrane space is the P side over there. So obviously ATP is produced in the cytosol only. Okay. Here is the summary of both the processes. This is an entire plant cell. Let's take it as a photosynthetic cell only. So their mitochondria is also present. So electron transport chain is taking place where ATP is synthesized, and in the chloroplast, the photophosphorylation is taking place. So this is by the photons, the energy that it comes is from the photons or the sunlight. And in case of oxidative phosphorylation, the energy is from the chemical bonds. The end result of oxidative phosphorylation is only ATP, but the end result of chloroplast is sugar. The ATP which is generated in the light reaction, it is anyways used in the dark reaction. So I can say that the end product over here is sugar, but yes, some of ATP is also released. So both will be generating ATP. Let's look at the differences now. Very important table, this one. If you want to learn such comparative tables, then you can refer to Biotechnica's book, a special book which is only for comparisons of processes like this, called as the concept table book. It will be available for the students to access. If you are interested, you can contact our team. Okay. Oxidative phosphorylation, it occurs in mitochondria, whereas photophosphorylation, it occurs in chloroplast. For oxidative phosphorylation, oxygen is reduced to water at the end. But as I told you, in photophosphorylation, we start with the oxidation of water to release the electrons and oxygen. In oxidative phosphorylation, electrons donated by NADH and FADH2. But here the electron sources from the water, that is photolysis of water. Oxidative phosphorylation, oxygen is the final electron acceptor. Here the electron acceptor is NADP, which is ultimately leading to the form of NADPH required in dark reaction. Okay. Here in oxidative phosphorylation, only ATP is synthesized, but in photophosphorylation, as we saw, ATP and NADPH both are generated. Oxidative phosphorylation is independent of light condition. It can happen anytime, but photophosphorylation, since the energy is coming from light energy, a light source, that's why it happens. It needs to happen in light conditions. Oxidative phosphorylation will be occurring in all aerobic cells wherever mitochondria is there, but photophosphorylation can occur only in photosynthetic cell. Finally, oxidative phosphorylation, the chemical energy is converted into ATP, whereas in photophosphorylation, light energy is converted into ATP. Okay. However, there are some similarities which you should know. First is both processes involve the flow of electrons through a chain of membrane-bound carriers. Then this free energy which is made available by whatever source, okay, the electron is, whenever the electron transport is there, the downhill exergonic flow is there in both, which is coupled with the uphill transport of protons. So one releases energy, one utilizes the energy. Both involves a proton impermeable membrane. The membrane name is different, location is different, but both are proton impermeable, and that is how they conserve the free energy of fuel oxidation. And the last point is that there is a transmembrane flow of electron down their concentration gradient through specific protein channel which provides free energy for synthesis of ATP. In both of them, it is catalyzed by ATP synthase, which is a membrane protein complex that will couple the proton flow to phosphorylation of ATP. I hope this video was helpful to you. Let me know in the comment section whether you liked it or not. Thank you everyone.